Hello and welcome to another tutorial for BIOS E1A. In this tutorial, we'll work through a practice problem which reviews what you learned from lecture about transcription and translation. In the process, we'll get some practice at going from a given DNA sequence to the corresponding mRNA sequence and to the matching polypeptide sequence. Let's begin with part A of the problem. Here, you're asked to take a given DNA sequence for a gene and to give the messenger RNA, or mRNA, sequence that would be generated from that gene. From lecture, we know this conversion of DNA into a messenger RNA is called transcription. But how do we take a given DNA sequence and transcribe it into an mRNA sequence? To answer that, let's look back and walk through the transcription example from lecture 2.3. Here we have an example of double-stranded DNA sequence, and we're going to transcribe some of it. We're given some starting information to know where to start transcribing. The +1 position, aka the first nucleotide position that will get transcribed into mRNA, is marked. We're also given an arrow icon that shows the direction in which the DNA sequence will be read and transcribed. So here, that's starting at the +1 and going to the right. The blue box marks the example gene that we'll transcribe. The two strands of DNA have different names based off of how they're used in transcription and how they compare to the messenger RNA transcript. The template strand is the strand that runs 3' to 5', in the direction transcription proceeds. You can see this in our example here, where transcription is running left to right, making the lower DNA strand the template strand. The template strand is the one that will be read by RNA polymerase when it's making the mRNA. The upper strand that's running 5' to 3' is the coding strand. As we'll see again later in this example, the coding strand has the same sequence as the mRNA made by transcription, except, importantly, the thymines or T's in DNA are uracils or U's instead in RNA. The two key points to remember about how the enzyme RNA polymerase transcribes DNA into RNA are that the RNA strand is made by RNA polymerase and it grows out in the 5' to 3' direction, and RNA polymerase knows what RNA sequence to make by reading off of the template strand of DNA in the 3' to 5' direction and doing complementary base pairing. If you still don't totally understand what that means yet, don't worry. We'll walk through this example and then part A of the problem step by step. First, let's copy our 3' to 5' template strand here. Our mRNA will be synthesized 5' to 3' off of this template strand, starting with the first base from the +1 position. First base in the template strand is an adenine, an A. In DNA, this would pair with a T, but we're doing transcription and we're making RNA. And RNA doesn't have thymine and T's, it has uracil and U's instead. So the A will pair with a U for the first nucleotide of the mRNA. Continuing to read 3' to 5' on the template strand, the next base we encounter is a T. T base pairs with A, so the second position in the growing mRNA strand will be an A. Then, keep reading 3' to 5' on the template strand to the next base over. It's a G. Guanine base pairs with cytosine, so the next base in the mRNA is a C. To finish making your RNA sequence, you can just keep repeating this process in the same way. The next three bases, 3' to 5' on the template DNA strand, are AAT. A pairs with U, A pairs with U, and T pairs with A. So the next three bases in the growing mRNA will be UUA. And again, the next three on the template strand are TAC, which will base pair with AUG as the next mRNA bases. For practice at doing this, I'd urge you now to pause the video tutorial for a second and continue generating the rest of this example mRNA using complementary base pairing yourself. After you do this and you carry on with the same process of reading along the template strand from 3' to 5' and generating the 5' to 3' mRNA by complementary base pairing, this will be the final mRNA sequence that you end up with. Walking through this base pairing one nucleotide at a time can be a little laborious, so I wanted to reiterate a point I made before about the similarity between the mRNA sequence and the sequence of the DNA coding strand. I'll paste the coding strand next to the mRNA we just generated to show you what I mean. Look closely at their sequence and compare. What you'll notice is something that I mentioned earlier, namely that the coding strand sequence is the same as the transcribed RNA sequence, except that T's in the DNA are replaced by U's in the RNA. If you're asked in a problem to transcribe the mRNA for a given DNA sequence, if you're given the coding strand sequence, you can use this fact as a shortcut for generating the mRNA sequence without the more laborious one-by-one base pairing we just did off the template strands. Okay, with that review of the lecture example out of the way, let's jump back to the practice problem part A. Here you're asked to transcribe DNA into RNA, but you're not given both DNA strands. Instead, they only give you one strand, the template strand. How can we use just the template strand to do transcription for part A? Well, remember what we know about how RNA polymerase makes use of the template strand. It generates the mRNA 5' to 3' by reading along the template strand in the 3' to 5' direction. That means we need to pay attention to the direction of our template strand. It's given in this problem in 5' to 3' orientation, at least reading from left to right, which is what we usually do. So the simplest way for us to read it and transcribe it to mRNA here is to flip it around so it's running 3' to 5'. Then we can read along it from left to right using complementary base pairing to generate the 5' to 3' mRNA. So let's paste the reverse template strand in 3' to 5' orientation here, and then we can transcribe it into mRNA exactly as we did in the previous example, by walking along the template strand sequence from 3' to 5' and doing base pairing. The first mRNA base will be a G pairing with C in the template strand. Moving 3' to 5' on the template, the next base is a G, so that base pairs with a C for the next base in the mRNA. Continuing the same way, T will base pair with A, another T will base pair with A. The following A will base pair with U, again remember it will be U, not T, in mRNA, and C will base pair with G. At this point, again, I strongly recommend pausing the video tutorial briefly and getting more practice in at generating RNA like this, by doing the base pairing for the rest of the sequence to finish off this mRNA. I've continued the process here and generated the rest of this mRNA by complementary base pairing. This RNA sequence, then, is the answer for part A of the problem.